you all for being here. Our next speaker is Dr. David Kem. I was really excited when Dr. Kem agreed to come to speak at the conference this year. He has been working on, as many of you know, different autoantibodies in POTS, and this has opened up sort of a whole new world of research. And as you can see from other presentations, this has really spurred interest in this type of research from other research centers as well. So Dr. Kem is a professor at the University of Oklahoma College of Medicine. He's a very, very respected researcher in endocrinology, metabolism, hypertension, and how these conditions affect the heart. His lab has been working on these antibodies in collaboration with Vanderbilt, UT Southwestern, and Dysautonomia International. So please give a warm welcome to Dr. David Kem.
Thank you, Lauren. It's a real pleasure to be invited to speak to this group. It's probably fortunate at one level that POTS affects women predominantly. Whenever a section head wanted to get something done, I assigned it to the women in my section. And I'm impressed with what the women can do in volunteering and getting things done. My wife is of a similar vein. Second, in terms of impressions, is that I find that in this current age, many of the people with a disease often know more, at least in details, than the physician they talk to. With the advent of the internet and search organs. So I'm sure that many of you are familiar with some of the issues that we've raised in this area, and I look forward to discussing them with you. Just as a beginning, you all know about the common symptoms. I'm really an endocrinologist and came into POTS through my interest in atrial fibrillation and control of tachyarrhythmias, rapid heart rates. And next to my office is a woman, a physician, and a cardiologist, and she asked me to see several of her patients that had a tachycardia. But as you know, cardiologists love to put catheters places, and it wasn't indicated for these patients. So he asked if I'd seen him, and so I said yes, because I had seen autonomic orthostatic hypotension patients, and especially diabetics in the course of my career. And it intrigued me when I saw the pathophysiology before it and made some hypotheses, because I only like to treat patients that I know a little bit about and what we're treating with. And that obviously was something that we needed to do. We've specialized in the cardiovascular aspects of POTS, and I think our work is relevant to these other areas. In fact, I'm certain it is. But the data that I'm going to share is primarily in this area with regard to the cardiovascular. And that's fortunate because we can measure that much more accurately than, say, gastroparesis. How many times do you throw up in a day versus what is your heart rate? And so this is an area that we have worked on. And as you know, there are many different attributed causes of POTS and their associations, and this is not complete, but it shows you that we have a very broad area, as has been emphasized, and that something can be learned from some of these. We've done a lot of work in the thyroid aspects of cardiac disease, and that's where we got into our autoantibodies that have been translated on it to the areas that we're working on. Autoimmune. What is an autoimmune disease? Well, this is something that most of you already know. But there's been a whisper, and we think now a shout, that a variety of autoantibodies may be temporally related to POTS. Now, you know that I'm not saying that they cause POTS at this point, but I'm going to present some data that I think is pushing us toward that. It is not yet clear that these are causative, however, and we want to go further. And we are going to provide preliminary data that the removal of these antibodies may support a future that is more hopeful than we have at the present time. What is an autoantibody? Well, autoimmunity is the system of immune responses of an organism against its own healthy cells and tissues. This is straight from the dictionary. Any disease that results in such an aberrant immune response is termed an autoimmune disease. That's pretty straightforward. Thus, there are hundreds of autoantibodies that are known, and not surprisingly, therefore, there are hundreds of autoimmune diseases. But why should we suspect that POTS is an autoimmune disease? Well, you've obviously seen the predilection for women who have a great majority of autoimmune diseases. Not to exclude men, but nevertheless, it's 5 or 6 to 1 or 9 to 1. It depends on the illness and the demographics, the age of onset, predominantly during the younger ages, but also in older ages where the immune system is altered also. It's associated with stressful events. It has a cyclic, but not daily activity in terms of the overall process, and it is importantly, it's associated with other autoimmune diseases. Why isn't POTS considered an autoimmune disease? Well, the evidence hasn't been gathered yet, and there are certain things that are used to determine if a disease is an autoimmune disease. Vitebsky was a physician who was interested in autoimmunity, and he made some postulates that are taken from Koch's postulates. Koch was a bacteriologist way back when, and he had some criteria for when is a bacteria or an illness caused by an agent. And Vitebsky changed those directly to antibodies. And some criteria that need to be met, and it isn't like there's only 1 or 2 or 5 of these, but that if you can show that a direct transfer of the antibodies to a model, usually an animal, because it's not considered wise to be transferring antibodies to a human at this situation, but there are situations where this is correct. Or you can have an indirect transfer. Induction of a disease by an autoantigen, that is the target for the antibody that is present in the body, and if you can induce the disease by using an autoantigen, that would be one of the criteria. Identification of autoantibodies are the T cells in a particular lesion. Well, POTS doesn't exactly have a single lesion along the line. It involves the whole body. So this is a little bit more difficult, but in areas along the line, if you can show that the autoantibodies are present in the body. So this is an area that you can see is obviously one that we're working at. Transfer the disease by lymphocytes into models. Again, going to the cells that put out the antibody rather than the direct antibody. Are there genetic models? A little bit harder. Circumstantial evidence, which is not as strong as these, but is considered important. Association with other autoimmune diseases, we've already mentioned that, and this is self-evident to some of you who are already walking examples of that. Association with HLA subtypes. These are related to the autoimmune systems and autoimmunity systems, and so that's certainly something. And a favorable response to immunosuppression, or that is to remove the antibody influence, and this is a very important one that we think is critical. What is an antibody? Now this is a cartoon showing an antibody. Obviously it looks different in the real tissue, but the important point is an autoantibody or an antibody has several fragments to it, a heavy chain, a light chain, and at this end and at this end, they're bipolar on this situation. They have a unique structure which matches the antigen that they were formed for. So this is the business end for identification of the antigen. Here, down here on this other end, there are some areas that interact with interferon and with other enzymes. And so this is, in effect, the hatchet part of it. This is identification, localization, and this is an area that interacts to cause things to happen, all the way from removal of the antigen to removal of the cell that's offended or whatever. But this is a remarkable construction. And it has certain areas where there's a hinge up here where this can vary in terms of its spread, and there are some other aspects. It's interesting that enzymes can be used to clip this part from this or this from this. And these have their use because these, in effect, are bipolar, as I mentioned. They can attach to two different sites that they tend to be the same. There are constructions that might be different, but normally they're the same. This is a cartoon showing a typical G protein coupled receptor, and I'll be descriptive. The issue is that the receptor is that snake-like thing. There are 7 transmembrane segments, and these are important, but they're not them in terms of bringing about an action. But the identification is in those little loops up above and the end of it. And the first loop on the left, second loop in the middle, third loop on the right extend above the plasma membrane and therefore are accessible to the serum, blood passing through, say, in a blood vessel. And we think that's important for the cause. Associated with this, going from right around the circle, is that when a ligand, which is norepinephrine or something else, binds to it, it fits into a cup, into that intramembrane area. It slides in, attaches, and electrostatically, that is magnetically, if you will, is attracted to certain areas, and when it lines up, it causes a change in the conformation of the lower part of the receptor, which brings about action. And therefore, when we talk about a ligand like norepinephrine, that yellow ball going in there and join it, that's an orthosteric effect. And it causes the changes in what we call G protein coupled. Well, the G protein is that little complex at the bottom. Does it work now? Mm-hmm. Thank you very much.
You're welcome.
Great. So at this point, you can see that this G protein then separates. That alpha, beta, gamma proteins are together. They get separated. The alpha comes over to an enzyme nearby and acts on it. And then, as a protection against too much activity, an enzyme clips off a high energy segment here. You can see it coming off. That's a phosphorus group. And it then dissociates and comes back to here. And this change in the effector is finished. Furthermore, other things are happening, not shown here, which cause this to be phosphorylated, and it gets rid of the norepinephrine. It changes the configuration with this other high energy, and the norepinephrine comes off, and everything returns to normal. So this is a normal cycle. It's like driving a car. If you push on the accelerator, it goes forward. But if you don't have a brake, you're in trouble. And this is the brake to slow it down and bring it back to normal. So this is an exciting concept that is occurring. What happens is that there are antibodies that are known to form attached to this first or second and even the third loop. And these antibodies can alter this configuration also, with or without the presence of the norepinephrine ligand. And this is where we're going to go now. Our hypothesis was that the roles for activating autoantibodies in POTS, and we think in related diseases of some, that the cause of it might be caused by, or the etiology might be caused in part by the presence of one or more of these circulating active antibodies. I've abbreviated them AAB. These may act as direct or partial stimulation. They also appear to alter the activity of the natural ligand, and this is very important. We think now that a major component is to change the receptor configuration such that the natural ligand, like norepinephrine, but it could be acetylcholine or other areas that are active, are either more active or less active on that receptor. So it isn't like the antibody is stimulating it, although it can, but that it's altering its relationship to its natural ligand. A unique cluster of these antibodies in each individual will alter their autoimmune system and produce differing but perhaps predictable signs and symptoms. So this helps explain why a patient might have the postural changes. That's what I'm going to show you. They might have as what the muscarinic effects that might affect more of the GI tract or the brain. And if a person doesn't have much of that, that might be a factor as to why they have less apparent disease in that regard. So that it helps explain a spectra of disease. We think, and this is hypothesis now, but there's now reason to believe this, that this is my attempt at a drawing of a G protein coupled receptor. The three loops are right here, and this is the membrane. And we think that enzymes that cause proteolysis, that is breaking down of the protein, like matrix proteinase 10, or equivalent endoproteinases that might be activated by a virus or some other activity, will come and cleave through the blood one of these or more of these loops out through their proteolytic activity. This loop is then processed, the extracellular loop, processed through the proteasome, which is an organism, not an organism, an organ particle in the cell that's sort of like a garbage disposal unit. Put stuff in the top, it gets ground up, and out comes the bottom parts. And it takes a bigger protein like this and turns it into 5 to 6 amino acid sequences. And this is probably 90% of your proteins in your body are processed through the proteasome. There are other means, but this is a very important one. And then this, for reasons that we don't know, are being processed through the immunoactive area to form antibodies. And these antibodies then go through B cells and T cell, B cell, or dendritic cells to form the antibodies. We think that this is a potential explanation for this. And there is a recent paper on Lyme disease, a different issue, but on the arthritis and Lyme disease, in which they did some processing to show that this equivalent, not with G protein, but with other fragments from the lining of the joints, could be converted into antibodies by this enzyme and could cause antibodies that were then identical to the ones that were found in Lyme disease in their patients. This is a very important concept that helps, and our laboratory is going to be pursuing this concept with regard to the proteasome. These are another view of the loops as they come up, and it's just a little bit more detailed than my pathetic drawing at that time. Now, this is important. I don't know if you can see it, but basically what it shows is the processing from the dendritic cells into the T cell and then the B cells and plasma cells to form the antibodies that are present and that we're finding in POTS patients. And this is just an illustration of the type of binding. Ignore this one, I just couldn't get rid of it. Showing the binding to this loop right here, the second loop, which is more common than the first. Normally this is just linear. Normally it'd be three-dimensional and there'd be a cup in here and the like. And it appears that when the normal norepinephrine comes in, this flops over to help trap it in there temporarily, and then it can flop out. With the binding of the antibody, it obviously changes this activity, but it also changes the structures down here. A Nobel Prize was given a couple years ago to a doctor on the beta-2 receptor, used that to show that just a 6-degree change in the angle of one of these intramembrane areas was sufficient to cause changes in the activity of the receptor. And so this is what we think is an important concept. Secondly, it's known that if you break this apart like this, they don't have any significant activity. And it suggests that the binding of this to another site, equivalent to another receptor, and the activity on that may be necessary for its action. Very complex. Okay, let's move on. In 2014, we described 14 patients. This is the reference. Many of you probably, it's open literature and you can pick it up easily. And in this, we showed that the IgG that was purified from the serum from 7 patients from our institution, and then we invited 7 to be, or more, they were blinded to us from Vanderbilt, were sent to us. And these showed that the IgG contracted arteries in vitro, and that when we blocked them by the alpha-1 antagonist, prazosin, that we could partially reverse, mostly reverse this. So in other words, there was alpha-1 receptor activity and in the sera from the patients with POTS. And the normal subjects had minimal effects. Secondly, beta-1 and beta-2 transfected cells that we had purchased and had a reporter in it that could be tested, and they activated the sera from the patients, also activated these cells in vitro, and was blocked by appropriate blockers, propranolol or a specific one for beta-2. And then, importantly, the IgG from these patients shifted the phenylephrine, which is a norepinephrine-like agonist, it's easier to use, and it shifted the curve, a dosage-response curve, to the right. What that means is that it was less effective in these cells when it was in the presence of serum from an IgG POTS patient. And on the opposite, the same IgG on cells that had only beta cells in it shifted the dose-response curve to the left, and the controls did not. In other words, they were more efficient, so that they were more active in the presence of it. So we have the suggestions that these antibodies could be related to the pathophysiology of POTS, and that we needed to do more studies. And it's clear that if we can show an activation of the antibodies, removal, it would be necessary to have proof of causation. I'm now switching to our recent study that's going to be published in the next 2 or 3 weeks in the European literature in Europace. And I'm going to show you that data, and it basically confirmed our first paper. We had 7 patients. Well, this is from the first study. This is our first study. And what we showed was that there was a dose-response curve for the alpha activity, and this is necessary to show it. And this is from the serum, and this is from the IgG to show that they were comparable. And then this is for the beta, also, that we had a dose-response curve. So this was to help validate that data. This first paper, and I'm showing you the data here, that the controls had 100% activity in that cremaster artery, and the patients from University of Oklahoma and from Vanderbilt both had increased activity in that contractile. And these are the group data here to show that in fact that they were active and could be blocked. Secondly, on the beta 2, these are the cell data, normal, abnormal. You can see that there's some cross-reactivity with the beta 2. The beta 1 group were clearly different all the way, and so we felt it was more important for the beta 1 than for the beta 2. This is the alpha data, and these are the patients that we had, and you can see that whether they had IgG alone or with it, with the controls didn't really change. They stayed about 95%, while the ones that had patients with POTS, they had a significant contractile in the green, and then when we put a normal blocker in for the get rid of any beta effects that could be interfering, and then added prazosin, they were tended to come up much more dramatically in the blue toward normal. So this was the data, these were the data that we were showing at that time. And importantly, this is that dose response shift that I was telling you about. The alpha takes when we added the IgG from the patients with POTS, it shifted the curve to the right. And you can see that this, what we saw log, the higher the number of -5 is more than -8. And so it took more drug to shift. So it was less effective. And then with the beta, it went just the opposite direction. And this is an allosteric effect, we call it, where it's the direct effect of the antibody on the receptor, but not directly on the, by blocking or enhancing directly the norepinephrine, but the norepinephrine or whatever was more effective or less effective in that presence. And so this was very important from our viewpoint. We then have got this large number that some of you may have donated blood 2 years ago, and we've assayed them twice actually. And we've had problems with this that we hadn't anticipated. We couldn't use the alpha cremaster artery. That's a very tedious, you get 2 or 3 done a day, and you can imagine how long it would take to do that. So we assayed these sera and then had to purify, IgG, and that would have been a major impediment to our very small lab. We used the alpha assay that was new to us, that isolated cells, and we thought it would be just like the beta. And in that we can do about 30 individual samples on a given day, and the beta assays were the same. To our dismay, we found no significant difference between the putative POTS and controls when Satish broke the code. We didn't know what they were when we were being run, and that was, of course, distressing. We then found out, using the sera at that time, that when you take the assays that we use for this alpha, that it was not sensitive until 10 to the 9th. That's what we expected. And we didn't think that any endogenous catecholamine would be a problem. And then when we added an enzyme that gets rid of any catechols, we had added this catechols by ourselves. When we added catechol methyltransferase, it eliminated it. And then when we put in the norepinephrine, same concentrations with sera, you can see that it was sensitive all the way down to 10 to the 12th, 11th. And this was in an area that was going to interfere with the samples. And when you get bloods drawn like you had them drawn with a needle, norepinephrine catechols go up immediately. And so they're very significantly elevated, and we found evidence that these were present in the serum as well. And then when we added the catechol methyltransferase, this was then the residual antibody in this serum left. And so it looked like we could measure it with the catechol methyltransferase in large numbers. And the company was unable to duplicate the COMT, and we were unable to get more. So we've recently had to go through very tediously purifying all the IgG, and we're just turning over the beta data when I get back to Satish, and he'll look at them to see what happens. The second study, the one that's coming out in Europace in the next week or two, we did 17 additional POTS patients in association with Dr. Fedorowski at Sweden, where he sent us these samples, blinded our guy that runs the assays. And these data are slightly different in that we use the new assay, and these are the alpha assay here, and these are patients with vasovagal syndrome, which we used as a positive control. And these were patients, that is to say, they had a disease, and these were normal controls. And you can see about half the patients were abnormal, but others cross-reacted. But when we gave specific blockade, their values came down toward normal, while these were higher. So it tells us that these did contain material that could be blocked, the antibody that was being blocked. This is the beta 1, and this is the beta. These are the beta 2 data. And you can see again, there's crossover here, and we didn't get the 100% area that we thought. But this has led to additional problems and work in our lab. When we've done the patients that were positive, we found that they still have a curve for the alpha that shifted to the right. This is just a single patient, but we've got several others that were positive in it. And for the beta 1, they shifted to the left. And some of these were patients who didn't have any direct activity. So we now, okay. So we've then gone ahead and redid the arterial contractility and confirmed it. So the other people have done associations. Steve Vernino has already shown you their studies with this group of muscarinic. Gerd Wallacott has reported 90% of his patients, the 56 POTS they were studying over in Berlin, have antibodies to beta 2. He didn't show the beta 1, but a lot of them had M2. And of interest is a pain receptor. And angiotensin was only seen, and they reported only one patient. We have trained 12 rabbits to be models for POTS. And I had pet rabbits. I like rabbits. And we've trained them very well. And we put them in a tilt table that has, they love it when they jump in. We give them apple slices as a reward when they get into it. And so they just calmly take it and wait for their apple slice at the end. We then immunize them with the target peptides to form alpha 1 and the beta 1 receptors. And we've done this before, and they get antibodies, and it makes them, in a sense, models for the POTS we're talking. After 6 weeks, we repeated the study after measuring their blood pressure when we raise them to 30 degrees slowly, so we don't frighten them or anything like that, and then to 60 degrees. And they're in their cage. Rabbits like to be in a tight area. They feel most secure in their burrow. And their heads are above, etc., unlike, but they're slowly brought up. And these are the data that this is the compound that we've invented, just as a background for this. This is called retroinverso D-amino acid. There's two types of amino acids, L and D, left and right. Our bodies are all L. Bacteria and others have the D amino acid, and they don't get metabolized by the body. The body can't do it. So they stick around, and we use this technique to make the target. This is a target for the beta 1 right here. This is proprietary. We've applied for a patent. for it, and so I can show it now because it's public knowledge that this is what we've applied for. And we've shown that in 3 different experiments for atrial fibrillation and hypertension, this effectively blocks these antibodies, and it's very specific for them. What we've done, I can't show you the data because it's not been published yet, and if I do that, they say, oh, it's been published, and we won't publish it. But I can describe what happens. When the rabbit goes up, their heart rate goes up about 45 beats per minute. Rabbits have a faster heart rate to begin with, and they go up with the immunized much more than they do in the non-immunized, which is only about 20. And then when we gave them our RID peptide intravenously, in this case, but we think it's going to be oral too, it blocked them by half. In other words, their heart rate was cut in half. And this was significant in our group. Now we're setting up more detailed experiments to look at this, but I think this is very exciting, and it gives us the anticipation that we will be able to block more than one antibody with these constructs, and hopefully with an oral tablet that doesn't even require an injection and that's tight. And this would be specific medicine, and we see this as a possible future for POTS therapies. Not the only one, but some there. Witebsky's postulates, I mentioned before. In the course of our studies, we have shown induction of the disease by the autoantigen, the X. We have shown identification of autoantibodies in the lesions, that is the patient. We have also shown an association with other diseases. That's not hard to do. And now we feel that we're showing a favorable response to immunosuppression. What we have learned, POTS is associated with a high degree of autoimmunity. 2, we find an association of cardiovascular-oriented autoantibodies with the cardiovascular manifestations of POTS. 3, we have preliminary data to support the concept that there are antibodies that may be causative. 4, we have invented a specific decoy peptide construction that permits us to acutely decrease the concentration of these antibodies. And we've also measured the antibodies, and they do decrease. In an animal model of POTS, we love them. And this is associated with a decrease in the cardiovascular signs that we're measuring in these models. And as I mentioned in 3 previous models for antibody-related diseases. And finally, others have confirmed and extended these observations to include other receptors that may be associated with manifestations of POTS. So I'm going to stop at this point and open for questions. I have a little bit of hard hearing, so shout it out.
How do we keep track of whether this comes out in the future for POTS patients?
Well, I would read the newsletter that Deanna. I'm totally, Mutual Admiration Society. She's incredible and is very, and so is Alan, just to put it out there. But the point is that they report these things, and I think that that's an excellent source for doing it. Now, as I say, these are, Google is amazing. It's prejudiced, it's got problems, and other search engines also can be used. All you have to do is put in POTS and autoantibodies, and you'll find very little right now, but this second one should be out in just a week or 2. And that would be the best way, I think. Yes?
In your whole slide about the process of forming the autoantibodies, what activates the MM10 to go and cleave the...
We don't know, but we think that there are a number of things. For example, viruses. Epstein-Barr virus is notorious for another cause for it, and there are a number of mechanisms that activate it. Usually it can be a cascade, it can be a calcium mediated or whatever. But that's one of the things. the whole list of stress, et cetera, are possible.
And this will be our last question, I think, because we got to get to lunch.
Thank you. Are there any POTS patients, any subpopulation for whom you would consider low-dose immunosuppression, say with low-dose steroids?
Well, you asked the question that I didn't show the data on, and it's a very important one. And that is there a room for autoimmune suppression? And we know that there is. There are a number of anecdotal reports about people using IVIG and fewer with subcutaneous and even fewer with plasmapheresis. And rituximab and other immune suppressants have been used in desperation along the line. And they are variably successful. We're doing a study right now that we're having 12 patients for 12 months on IVIG. It's already in place right now. And we have one patient who's going through a year. And I can tell you that within 1 month of IVIG, her beta dropped with 1 month. Just 1 month of therapy. And yet it took 3.5 months or 4 months before her alpha dropped. So it wasn't just a dilution or something like that. And that's roughly proportionate to some of the improvement that this young lady showed. And we did our first assay, which is the only one we've done. We called everybody that we had up to date. We found that 4 of the 5 had shown a drop in their titer or the activity of their antibody, and they had improved, and 1 that hadn't improved didn't. So we think that we're hoping to take this to the companies that manufacture it, try to get additional support, because it's an incredibly expensive proposition. But what we would like to do is to get them to say, hey, and insurance companies particularly, when we had this one patient, we dropped her concentration from 1 gram per kilogram type of thing down to half, and she did fine, continued on like that. We also then went to 6 weeks instead of 4 weeks. That didn't work as well. And so we're also doing direct studies to see, to show the impact of the IG on the actual antibodies. And we just had very preliminary, and we were able to show that it suppressed it, but at higher concentrations than we really are relevant. So we're excited about it, and we are very appreciative of the initial support that DI has given. And we've recently gotten a larger support from the NIH for this, and we're hopeful. It gives us a guarantee of 3 more years.